Good afternoon, everybody. I really have the honor to introduce Hibba. Hibba has been working in the Aquaponics lab, and in this project, she was in the capstone undergrad. So she was instrumental in not just putting systems together, but organizing protocols, SOPs, the lab in general, advising undergraduate students as well as graduate students. And the most commendable part is that she has been doing all this research and also working full-time as a technician, lab technician in Dr. William Snell's lab, and in a topic completely different to this one. I'm going to read about it because it's interesting. They use green algae, Chlamydomonas, as a model organism to study and understand cell fusion and adhesion mechanisms. Currently, she's wrapping up a project. It's writing a paper with their collaborators in Paris, and it's very excited about her first publication. Hibba, thank you very much.
Thank you so much for the introduction, Dr. Azursa, and thank you everyone for joining me in my seminar presentation today. So today I'll be going over assessing the impact of nanobubble treatments on aquaponic systems. Special thanks to Dr. Azursa for being my advisor, as well as my committee members, Dr. Miller and Dr. Shakely. Also, I would like to first take a brief moment to give a shout out to all the undergraduates that has helped me throughout this project. Could not have been done without your guys' help, so I really, really appreciate everyone's help. So just a brief outline of what I'm going to discuss today. So we're going to go over the relevant background, research objectives and questions, some literature review findings of a review article that I'm writing with Dr. Azursa, experimental data and results, some challenges that I faced along during this project, and then key takeaways. And then the floor will be open for questions and feedback. So first, I would like to start off with going over the basics, and that is looking at the problems. It's not working anymore. Will it turn off? No. Okay, that's okay. No, it's there. Okay, so first, I would like to go over the state of agriculture production in the United States. So the main problem or challenge that we face is we need to produce more food, and that is to keep up with the growing population. But that's all to keep in mind when we've seen a 14% reduction in cropland. We need to increase agricultural production, but our rural farmer labor force is actually decreasing, and in 2022, it only accounted for 1% of the population. Other than that, our total energy use when it comes to from the food to transport it from one place for another accounts for 14%. Apart from that, 10% of greenhouse gas emissions comes from the agricultural sector, and then there are also issues with regards to freshwater resources because of climate change. The next topic that I would like to bring our focus to is the current state of food security in the United States. And so this data is from the USDA. In the year 2023, about 14% of households were food insecure. And then if you look at children, how many households had children that were food insecure, that number stands at about 18%. So then we can look at aquaponics as a solution to address both the agriculture problem and the food security problems that we're facing. So aquaponics reduces water usage by 90% and energy uses by 70%. It reduces the input of chemicals such as fertilizers because aquaponics uses the nutrient water as a source for nitrogen for the plants to grow. And as well as it reduces carbon footprint and cost. So essentially you can have this set up in urban city, in empty vacant buildings, and so that would reduce the transportation costs and also the distance where the food grows for the consumer to buy. So aquaponics as a sustainable system. So here is one of the systems, too far back. So here we see, so this is the grow media systems that I'm using for my research project. So aquaponics essentially combines aquaculture, which is the cultivation of fish in controlled environments, with the cultivation of plants and with using the nutrient water that comes from the fish tank. And that's the hydroponics part. So now going over the main parts of aquaponic systems. So they're essentially, obviously you need the fish tank with fish in them, you need the grow bed with grow media and for the plants to grow, but then there's also a water pump used that could pump the water from the fish tank into the grow bed, and there's a decent number of plumbing that's involved. So going from the fish tank to the grow bed, and then from the grow bed back to the fish tank. And as well as microbes, these are the most important part of aquaponic systems, and they're usually found in the biofilter. In the grow media system here, the grow bed acts as the biofilter. So now how does the nutrient cycling works in aquaponic systems? So first, you have fish, so you can see there's one fishy here and there's another fish right there. So they excrete ammonia, which ionizes as ammonium in water. Then you have in the biofilter beneficial bacteria such as Nitrosomonas and Nitrobacter that convert the ammonia to nitrites and then nitrates. And I like to call the beneficial bacteria as the unsung heroes because you can't really see them, but they're the ones doing all the work. If something goes wrong here, then you're not going to have a balanced system. Your plants aren't going to grow. There's going to be too much stress on the fish. And so you don't want that to happen. Finally, when it's finally converted into nitrate, plants use that as a source of nitrogen to grow. And finally, you can see this plumbing right here that goes from the grow bed down to the fish tanks. So the recycled water goes back down here, hopefully with low levels of ammonia, nitrites, and nitrates. So even though aquaponics could seem very beneficial or it has its advantages, unfortunately there are some disadvantages. And I've alluded to some of those on the previous slide. One of the main ones is water quality and nutrient balance. You need to have a good nutrient balance or else ammonia, if there's too high amounts of ammonia, that could be toxic to the fish. As well as you need to make sure there's good levels of DO, dissolved oxygen, for both the fish and the plants, as well as pH. Those two variables could depend on the species of plant and fish you're using. You need to make sure that they're in those ideal ranges as well as temperature. So then, given all these problems with aquaponics, we could look into using nanobubbles as a solution. And one of the reasons why we could look at them as a solution is because of their small size. It's because of their small size. I'll play that video. It's because of their small size that they're stable for a very long time in the systems. And so they follow Brownian motion, as you can see here from this video right here that was collected via the nanosite instrument when we were doing analysis of the water with the nanobubble treatment in it to detect if we can see the size and concentration of the bubbles in the water. And so they follow Brownian motion, and that's basically small random movement which allows them to remain suspended for a longer period of time. One other thing I want to point out, if you read the review articles or papers, there's generally some people define nanobubbles with different ranges. Some might do lower than 100 nanometers, some might bump it up to 200, but generally it's accepted that the gas bubble in liquid suspension has to be less than 200 nanometers for it to be considered nanobubbles. So there are other nanobubbles properties that are very useful for applications into aquaponics. So specifically, they can produce reactive oxygen species, short for ROS. And so what these can do, they can oxidize pollutants and pathogens in water. And so implying nanobubble-derived reactive oxygen species can inactivate harmful pathogens that might be spreading in your systems. Apart from them, the nanobubbles are known to have a negative surface charge. And so what that could do is they could essentially bind to positive cations in interface. And like I mentioned, they're known for their stability and longevity. So they don't rise, and because of Brownian motion, they just remain suspended in water for long periods of time. And it's because of all these different properties, they have a wide range of applications, from ag to hydroponics to aquaculture and even sanitation and cleaning. But if you notice one thing that's missing, that would be aquaponics. And so this was from a review paper. They went over a lot of different applications, but they did not specifically mention aquaponics as a potential field for application. With that, so there are several companies that generate nanobubble generators. One of them is Moliere. So that's their screenshot right here of their webpage. And as you can see, they really do want to market to farmers or growers to use their technology to in their systems. And if you just Google like how much it's cost, it's not cheap. It's a pretty decent investment you want to think about before you start applying these. And specifically for aquaponics, so like I mentioned on the previous slides, it's used for cleaning and sanitation. Well, for aquaponics, the main driving force are the microbial communities. You don't want it to do too much of a cleaning job that it get rid of those communities. And so if you're a grower who wants to invest in this, you want to make sure that it's really applicable to aquaponics. And that's why we're doing research, because there's really a lack or a gap in that field. So that leads me to my research objectives. So the first one is examining the current trend in research regarding nanobubble application and assessing the need for more research and any gaps that we've seen. Next is examining bubbles' impact on water quality. That includes the physical and chemical, fish and plants. Then here separately we have a research objective to look at the relationship between nanobubble treatment to microbial development. That includes bacteria, biofilm, and biofloc formation. And finally, examining the impact of bubbles on aquaponic components after 6 months. And so that's defined as a long-term time point. So from 6 months, so I've added the treatments 6 months after, are we still able to detect their impact in the systems? So here, because there's a lot of ground to cover, I'm only gonna speak about in this presentation, this text that's highlighted in black. So the one that's grayed out, I won't be able to talk about in this presentation specifically. With that, it leads me to my research questions. So I'm asking what trends are seen with the research of urban agriculture and nanoscience and technology, and how is the research gap between the two fields? Does nanobubble treatments have a positive impact on physical, chemical, so by chemical I mean the nutrients, water quality parameters, fish and plants? Does bubble treatment result in less microbial, biofilm, biofloc formation in aquaponic systems? And finally, what is the long-term impact of bubble treatments in recirculating aquaponic systems to after 6 months? Again, I'm only going to be focusing on what's in the black text for today's presentation. So first, I would like to briefly go over literature review that we've done. So the method that I've used Web of Science. We selected the date range for the last decade, so 2014 to 2024. And we had keywords that relates to urban ag, as well as nanoscience and technology variants. And so here you can see these are the key terms we use, and these are the respective graphs. So key things to take away from this, really since 2019 to 2024, across all three of these, we see there's been an increase in the number of publications that have been published. Similarly, if we look at the nanoscience key terms, we also see that a similar increase from that same time range, from 2019 to 2024. And here I'm showing the graphs for what's bolded. However, when you do a combined search, so here are all the options we had for the combined search. You don't really see the same kind of results, with the exception of aquaculture and nanobubble. Oh, sorry, aquaculture and nano, we have about 1,400. Hydroponic and nano, close to 1,654. But if we specifically look at what I am doing with my project, aquaculture, nanobubble, aquaponic, nanobubble, only 4 publications in the last decade, and then hydroponic and nano was slightly higher at 16. So this really indicates that despite the fact that there's an increased number of publication in urban ag and nanoscience and technology separately, there's not really that much publication with combining the two. So there's really a gap in the understanding of how nanobubbles could really impact on aquaponic systems. So that leads me to my research component. So when sitting down, mapping out all the different components of the research, obviously there's the design component. We created these systems from scratch. We decided to go with the grow media type biofilter with the internal bell siphon. For the treatment for this presentation, I'm focusing on nanobubbles and control. Control is just, it has no nanobubbles in them. Water quality parameters, so that includes the physical, and then the chemical, which is just the nitrogen content, so ammonia, nitrites, and nitrates. Fish health, behavior, physical, length, weight, microbes, so viable counts, what is growing, and then metagenomics. And finally, we're looking at plant health. So when I'm adding the treatments, the height, number of leaves, and then when we harvest the plant, we get the wet and dry weight, as well as chlorophyll. So from that, we divided it up to these experiments, 8 experiments right here. But like I mentioned, we don't have time to go over everything, so for today, I have data that I'll be showing for these experiments right here. And another note I would like to make, this part of the project is still ongoing. I've completed 2 harvest cycles, and a 3rd one is going to start this weekend. So for the experimental setup, so it's a randomized block design. Each system contains a 10-gallon fish tank and a 4-gallon grow bed with expanded share as the substrate or the biofilter. I'm using goldfish as the organism or fish in these systems. And one of the reasons why we decided to go with goldfish is because they're really sturdy. And with lettuce, we decided to go with lettuce because it's a commonly used crop for aquaponics. We had a water pump to pump the water from the fish tank to the grow beds. So we had 3 systems or technical replicates for the nanobubble treatments and 3 systems for the control, so no treatment added. One replicate that we're considering is like a whole harvest cycle, so that's about 28 to 30 days. And these in yellow, these are the tanks I'm using. So they're randomized. Here, this is the control, this is one of the nanos, another control. We have a nano here, control, and then another nano tank right here. So just very briefly, obviously before continuing with the research, we needed to optimize or see like how long we should be adding the treatments for. We can't have the treatments be added for, it's not like an air zone that you can turn it on and have it going on continuously because, and I don't show that data here, and we can, if you're interested, we can talk about that after the presentation. If you keep the equipment on, the yield is going to start rising, and you don't want to do that because then fish could get bubble gas disease, and you don't want the oxygen level to be too high for the fish. And so we did different time points, 10, 20, and 30 minutes, and decided to go with the 20-minute time point for our recirculating systems because that's a pretty good midpoint. And here, this is the part that actually is producing the nanobubbles. So on to the experiment, the method and the results. So for the fish data, we collected before and after weight, and so here's the setup of how we were measuring. So we collected the weight in batch. So there's 4 fish per tank, and so we just had this on a scale. We weighed the 4 fish, and then next to it, to the side, you see the bin with a damp tissue paper where we were collecting the length of the fish. And so in regards to the data, if you look at the nanobubble fish in terms of weight, it seems pretty consistent. They haven't really, gained or increased that much weight. at least for the first harvest cycle. The control tank's fish lost weight. We're not sure why that might be. But however, for harvest cycle 2, we see that both systems, we saw an increase in the fish weight. Similarly, for the length, we see like in both harvest cycles, the fish increased in length. Not more so in harvest cycle 2 than harvest cycle 1. And to the side, you see how we were measuring the fish. And so the top is from a control tank. This is from one of the nano tanks. And so these two data suggest that there is definitely a lot of variabilities with regards to adding the treatments to these tanks. With regards to the plant data, so as I was adding the treatments weekly for 20 minutes, each week we collected plant data in terms of height and the number of leaves. And so this pinkish color is for the nano and the purple color is for the control. Here you see, this is at week 4, what the nano tanks look like in terms of plants. And this is for cycle 2, week 4 in one of the control tanks. So I'm only showing you a picture for one of them, but there was a lot of variability between tanks. So we had one tank that the plants were slightly larger, and some tanks like this one, the plants were slightly smaller. So we are seeing a variability from tank to tank in terms of treatment. But if you look at the height data, they don't really vary that much. There's a lot of overlap in terms of like margin of error for both harvest cycles. As well as for the number of leaves. Nano tends to be, when it comes to like the second harvest cycle from week 4 and 5, it's slightly less number of leaves. That wasn't the case for harvest cycle 1. Similarly, for the weight, here nano is slightly lacking behind control tanks, and for the height, they're roughly similar. Like in week 3, nano tanks were taller than week 4 compared to the control. So when it comes to harvesting, so we collect the wet weight and the dry weight. So if you look at the wet weight here, so this is from the control tanks and this is from the nano tanks, you can visibly see the difference in plant density. And so we cut the root right here to separate the shoot from the root. And so the shoot weight, you can see in harvest cycle 1, nano, the grow beds that had the nanobubble treatments were slightly lower than the control tanks in terms of weight. And similarly with the root, they were lower than the control tanks. And you can really visibly see that from these images too. The nano tanks plants weren't as dense as compared to the control tanks plants. And so we put them in an oven at 70 degrees C for 48 hours. And after that, we took the dry weight of the shoot and root. And so this is what they look like dried. And so nano tanks, there's slight overlap. And similarly with the root, it's lower than the control tank. So not as dense in terms of the control systems. So the next thing that we checked for was water quality, chemical water quality in terms of nutrient. So we use colorimetric assays using the Hawk DR3900. And so we tested for Nestler, nitrite, tested for ammonia using the Nestler method, nitrites using the deionization method, and candium reduction method for nitrates. And so basically, if there's any nitrites present, ammonia present, you'll see a color change in your sample. So right here is the control. So that has that's clear, that's our blank, that has nothing added to it. And so when we add the reagents, if there's levels present, we see a color change, as you can indicate here. The intenser the color, the higher the concentration of their respective components that you're looking for. So in regards to the data, harvest cycle 1, so we collected samples for both, we tested in grow beds water samples and fish tank water samples. And the reason why we did that was because we're adding the treatments to the fish tank, but we're hoping we're expecting to see an impact on the grow beds. So that's why we decided to test both water samples, the grow beds and the fish tank. And so in terms of ammonia, this is what the fish are producing. So it would indicate if the fish are stressed or not eating well, that would correlate to how much ammonia that's being produced. And so here, this is week, so it's a 4-week harvest cycle. And so here you see nano starts out a little bit lower than the control, but as the week goes on, there's an overlap here, so they become pretty similar in the grow beds. In the fish tank, we're expecting there to be higher values than the grow bed because the fish live. The fish live in the fish tank. However, if you notice, there's not that much of a difference between the grow bed samples and the fish tank samples. There is a decent amount of variability that we're seeing with these margin of error bars. And so this is for harvest cycle 2. Not really the same trend is being seen. So again, there's a lot of variability from harvest cycle to harvest cycle. Next up is nitrites. And so here we see in the grow bed, the nanotanks are really, it starts out a little bit higher, but then after week 1, it drops and then it increases, and then it drops and then it increases. The control tanks in the grow bed don't really see that sharp of increase or decrease. And then if you look at the fish tank water, we see that similar pattern, but really more so like from week 0 to week 4. It makes sense here, like week 4, we're detecting high amounts here, and so we should be able to detect somewhat higher amounts here. And then if you look at harvest cycle 2, we don't really necessarily see that same trend here. Both relatively start out similarly. Week 1 is a little bit higher, then week 2 it drops in the grow bed, and then it increases, and then it drops. Similarly with the fish tank water, starts out high, increases, then drops, then increases, then drops. So with regards to nitrates, this would tell us about the plants. If the plants are taking up the nitrates as a form of nitrogen to grow. And if you recall from the previous slide, I showed you the image of the plant from nanotank 4. Those plants don't really look that large, but there is a sufficient amount of nitrates present in the system for the plants to be taking up, but for some reason they're not. So here, nanotanks, it starts out as pretty high in nitrates, still stays high, then it drops, and then it increases. Similarly, this is a typo, this should say fish tank. We see that similar trend in the fish tank. In terms of the harvest cycle 2, starts out not as high, goes up higher, then drops, but still higher than control. Here they're relatively the same, but at week 4, nanotanks are definitely higher in nitrates, but control tanks are not as high, yet the control tanks yielded with larger plants as compared to grow beds. So in terms of the microbial data, we wanted to see what is able to grow using spiroplating methods. And so during the harvest cycle, there's not that much difference between nanotanks and control tanks. Similarly, in the grow bed, we're not really seeing that much difference between the two. They're pretty similar to each other. And then we also collect, one plant would go for microbial sampling on harvest day. Here we see in the leaves, the nanotanks are lower than the control tanks. But if you look at the root, both of them are pretty high, and that makes sense because a lot of microbial activity goes on in the roots. And then if you look at the rock wool and stem, they're still pretty similar. So in terms of turbidity, we also collected turbidity data. For harvest cycle 1, the nanotanks were lower in turbidity as compared to control, but you can say overall for harvest cycle 1, turbidity was low overall. Whereas harvest cycle 2, we do see it is higher as compared to harvest cycle 1, but we are seeing a lot of variability for both the nano and control. So in terms of turbidity, there's no clear trend with that either. We're seeing a lot of variability between the treatments. Finally, I would like to go over challenges. So I actually collected an initial set of data over the summer. Unfortunately, we found out that samples we sent for microbial analysis got contaminated, and so I had to repeat all 3 harvest cycles again. So I was in the process of doing that this fall, and then we had a very cold winter where with my second harvest cycle, the plants did not grow as well. And so we paused, we waited for the temperature to warm up a little bit. I just completed harvest cycle 2 last week, and we're gonna start harvest cycle 3 this weekend. So that was just some of the data. Like I mentioned, there's still a lot that's ongoing. So harvest cycle 2 is gonna start this weekend. We're going to do metagenomic studies on these, so water samples have been collected for that and kits have been ordered. We're also, the other part of the microbial analysis, looking at biofilm and biofloc, and so that's FTIR analysis as well as TEM imaging to see physically if we can see a difference between the biofilm that's growing in the grow bed and fish tank. And my committee member, Dr. Milner, took some preliminary samples, and we did see some differences in the physical structures from treatment to treatment. And finally, a major question is when, especially given the microbial data, it could be the method we're using isn't capturing the changes in the microbial data, or it's like the time point, because I add the treatments and after 48 hours, we're collecting the microbial data. So maybe between that time, the systems are recovering and we're not capturing the decrease in microbial population. But we need to ensure that as I'm adding the treatments over the course of time, the nanobubbles are still there. And with another lab, we worked on a ROS detection assay. So it uses fluorescein. It's a fluorescence assay. So you add fluorescein to nanobubble tanks, water, control tank water, as well as our positive control is hydrogen peroxide. It's known for producing a lot of oxygen radicals. And so we would measure the intensity and see if there are any radicals present, because that's an indicator of nanobubbles. So some key takeaways. So there's definitely a gap in the field. There's not much research being done in terms of nanobubble applications to aquaponics. And so that's why this research I'm doing is very important, is to close that gap in the research field. We could say it's like I guess not necessarily a positive impact, but it's definitely not a negative impact. We need to wait until we complete the 3rd harvest cycles to see which way it's trending. Obviously, they're not losing weight, so it's not a negative impact, which is a very good thing. And so, but we are seeing this sort of negative impact on the crop biomass. It could also be dependent on the type of species we're using. If you look into literature, some, like, this one paper used carrots and they saw a negative impact, but if they use another species, they saw a positive impact. So it could be species related. And I suppose the good news is so far we haven't seen an impact on microbial populations, which is good because the microbials are the driving force of aquaponic systems. And so if you clean them out, you're not gonna have a working aquaponic system. And so that's the end of my presentation. Questions?
Questions? I have a question.
Yes.
So I think it was on the previous slide, it was on point 3. It was, yeah, actually go forward a little bit. One more, please. Yeah, so nanobubble treated systems yielding crops with lower weight density. What's the explanation there? Do you have thoughts?
That's what we're trying. We thought it was a nutrient, but like as you saw, there's enough nitrates for the plants to be taking up. So we're not. Maybe it's some other, or it could be that because when we germinate the seeds, we germinate them with like plain, like we're not germinating them with nanobubble water.
Right.
So maybe when we put them in from a non-nanobubble treated system to a nanobubble treated system, maybe there's like they sense stress, and maybe it could be a stress response that they're small. Yeah. I mean, this is bringing in like molecular biology, sort of like, I guess we could, or someone could look at RNA-seq data to see maybe if like those transcripts are upregulated. That would indicate that's what's going on. But there's definitely enough nutrients for them to take up.
I was also thinking, well, so the total nutrient load might not be changing.
Mm-hmm.
But one of the things that could be changing is that the reactive oxygen species might be changing the organic
Mm-hmm.
material that's there, or something, you know. So it might not be the total amount of nutrients that is affecting the plants. Another possibility could be that it's like the chemical characteristics of whatever compounds the plants are interacting with.
Yes,
may
definitely.
be affected.
Definitely. Any other questions? Yes.
So the fish, don't they eat like some of the plant-related things, or at least the detritus? So how can the plants go down and the fish go up, or is there some other effect on fish?
We haven't seen that much of a negative impact on the fish.
I guess
We're-
how is fish going up? What's causing them to go up, I guess?
Well, they're going up in terms of here. I'm not sure why in the control tanks we saw a decrease in weight. Maybe it could be like a scale error. It won't be properly balanced that day. But here in Harvest Cycle 2, we see both of them going up respectively. I also want to make a note. Maybe this isn't like the best way to present this data. Maybe like doing a change in weight before and after. That might be more. That might tell us more.
Well, I guess I was more asking, why is the nanobubble one higher than the control? Why is the fish biomass higher?
So they're just larger fish like that. This is what this is the fish I started out with in these tanks.
Yeah, it'd probably be better to do the difference.
Yeah, difference. Yeah,
Yes,
it was only
okay.
after I made the slide I realized that, and then I didn't have time to go back and change it.
Yeah, because so off of that though,
Mm-hmm.
and I don't know if this is in there and it's just like a little bit muddied now because of like, you know, the starting weight or whatever.
Yeah.
But there's actually, I feel like there might be a mechanism to that where using nanobubbles might make bigger fish. to some
That's
extent,
what they're so.
because the oxygen would be more efficiently up.
They're
like, taken
supposed.
up.
There's a Now it's escaping my mind. There are different parameters that you can measure fish health by. It's a calculation that you like specific growth or daily growth weight, I believe. You can do that calculation and see. So the lower the calculation, the more efficient it is. So they're turning food into more body mass. And so we should, after like the completion of the third cycle, sit down and like, do those calculations for all 3 cycles and see if that's really being impacted.
Yeah.
Yes.
I saw you mentioned that you were doing like fish behavior.
Mm-hmm.
That was one of the factors you were looking at. Is that just like observational?
Yeah, observational. We have a lot of observational data for the fish. So when we're weighing the fish, we notice how they're swimming, if they're active, if they're eating well. We take a note on that. So that's all in like a spreadsheet and soon to be decided how to analyze all that data. Sorry, hand here, yes.
Me?
Yes, you.
So 2 questions. I'm
Uh-huh.
assuming you're feeding these fish.
Yes.
So the diet is based on percent of starting mass of fish or something?
2.5% of their
Okay,
weight.
so they get a daily diet. You mentioned you had some temperature issues. Did it get really cold before harvest cycle 1 and then warm up and get nice and toasty for cycle 2?
Could you repeat the question? I think the answer is yes. I just want to make sure I understood it.
I'm guessing they got cold and decided not to grow and just hung
Yeah, that
out for a
could-
while. And then it warmed up and they started thinking, hey, this is great, feels good, and they started eating more actively and
Mhm. That's a very good question. So this harvest cycle 1 was done November to December, and then this one was done in the month of March, so March, April.
But they don't know months, they
Yeah, I
know that
know,
it
yeah.
felt like it was becoming winter. And
Yeah.
then you got them someplace nice and warm and got a bunch of lamps on them or whatever, and they thought, oh, feels like spawning season, feels like, you know.
Yeah, no, that's a good point. I don't present this data here, but we do collect daily dissolved oxygen, temperature, and pH data with using a probe. That data isn't shown here.
And one other question, you said that the turbidity went up in your tank water for harvest cycle 2.
Yes, here.
And so this is the same water, same fish.
Yeah.
Some gap in time between harvesting and starting the next one and so forth. But my guess is all of this organic media that's now in the fish tank is ripe for interaction with the nanobubbles.
Yeah.
So you're right, that there's a lot more happening in there in this second one, good or bad, I don't know, but.
Yeah.
Right.
No, that's definitely a very good point. Any other questions? Yes.
So bubbles in a piping system or in a pump are bad. They'll cause it to kind of corrode and they'll cause your impellers to fail. Is there anything like that for nanobubbles? Like, will it increase your maintenance costs? Do you see any of that in the literature that you're looking at? Or no?
Not that I've noticed. We do clean, so between harvest cycle, we do have like that 1 week break period. That's for me to like go in, make sure all the pumps are working, the tanks are clean. And this is just like observational. We are not like collecting data on like the amount of biofilm on the water pumps, but the nanobubble pumps are cleaner as compared to the control tanks. There's not that much biofloc buildup in the pumps.
There are also some experimental anti-fouling systems that use bubbles. So yeah, cool.
Yay. Any other questions? Comments?
No other questions? What is a fish?
What is aquaponics?
Yeah.
What is nanobubbles?
This might be very dumb, but what is the nanobubble system supposed to do for the aquaponic system?
So that's a very good question. So nanobubbles is used a lot for like specifically for aquaponics, hydroponics, and ag systems to, well, I guess specifically for aquaculture to have like bigger, healthier fish, and then for the crops, bigger crops, essentially.
So more oxygen.
More oxygen, yeah, but we're not necessarily seeing that. These are bench scale systems, and so it is much smaller. Maybe in like a commercial systems, they might be a little different. Yes, Dr. Ursa.
This is not a question, but maybe I can help to answer a little bit because some nanobubbles are used commercially to sanitize products. That's what brings us our attention. Maybe that's gonna happen to the aquaponic systems if we use oxygen nanobubbles. That's what we use in this experiment.
Yes.
I guess what you mentioned that like the systems themselves were cleaner, like if you saw maybe, like if you end up seeing that there's like a negligible or even like a slight detriment to the growth of the produce, do you think that's worth like maybe having to do less maintenance to like clean the tanks?
Well, if you're a commercial aquaponics system, like you want to sell these crops to whoever you're selling them to, so you want to make sure like your crops are well enough so you earn your money. Cleaning the systems is another issue. Like there's a lot of biofilm and biofloc formation in the pipe systems they use. That's something for the growers to decide, or maybe it could, because I'm posting the system, so I'm only adding treatments 20 minutes a week. Maybe they want to do more or less depending on that, finding their own sweet spot. But yeah, that's a very good question. There's a lot more work that needs to be done. This is just scratching the surface. Yes.
Do the bubbles last, like,
That's
days
why
or weeks, or are they?
that's a good question. That's why we worked on. I just want to go. No, not that slide.
Well, I
to
know it's a
this.
remaining question, but does the literature suggest that if you generate these, they'll be there 2 days later?
In like stagnant water, they've been stable for a long time, but these are circulating waters with the bell siphon, and so the water builds up in the grow beds and then it flushes out, and so that is a disturbance. And nanobubbles are sensitive to disturbances that would collapse, causing them that disturbance would cause them to collapse. And so theoretically, they shouldn't be lasting this long in the systems, but
I got one last question slash.
no, these are very good questions.
Fish don't like bubbles. In other words, air. They like dissolved oxygen.
Yeah.
So the relationship, if it's staying in a nanobubble, it's not necessarily beneficial to them. It's that the surface area, so forth, that ought to get more effectively creating dissolved oxygen for them. So I'm wondering, you're measuring DO.
Yes, we are.
So you have that relationship. Once you get to a certain point, they don't need more. As you said, they can have problems with it.
Yeah.
Okay.
Was there a question or was that a comment?
Well, I'm trying to
You said
think about
another
this, and
question.
I'm thinking, okay, there are these little bubbles and they last, that's great. Then I'm like, but the fish, that isn't useful to them across that gill. They need it to be in a dissolved media where it's gonna make that exchange. Or am I wrong on that?
I think nanobubbles also increase the of oxygen. We got 22 parts per million of dissolved oxygen when adding nanobubble.
But that's
Okay.
only
And
temporarily
fish
when the.
like 8.
No, that was not in a fish tank.
Okay, that's in a sack. I was like, wow.
But also, Molia, the company that she mentioned, they claim that the nanobubbles can stay up to 60 days in the water, that's
Okay.
dissolved oxygen.
But they're not dissolved.
No, but
By definition.
they sell it because aquaculture needs dissolved oxygen.
Right. So they can continuously put in for a long period of time as opposed to a bubble that just runs to the surface. Okay. Interesting.
A comment on that I want to make. So once the equipment is on and I'm having it on for 20 minutes, dissolved oxygen continues to increase. When I shut it down, because I collect data at like the 1 hour time point and a 3 hour time point, as soon as I turn that equipment off, DO levels drop back to the control tank. So 5 to 6 ppm in that range. So that's something that I observe. It's only when the equipment is on is when it starts, the DO levels start increasing, but after it's off, it goes down and remains down during the week.
So I guess the sanitation aspect is probably the more important than the oxygen aspect of
Yeah.
the nanobubbles.
Yeah, that's a very good point. That's why in one of the slides I mentioned, like, it is used for cleaning, but we don't want them to do too much of a good cleaning job in these systems to eliminate the beneficial microbial populations.
One more question.
Yes.
For the microbial metagenomics, are you thinking about, in addition to like microbial community composition, are you also thinking about looking at the genes that are there?
That's a question for the person on my committee. Because all I know is kits have been ordered. I don't know any more details other than, I think it's 16S RNA method they're using, but yeah, kits have been ordered. But, because they're at the USDA and there are a lot of issues with the government right now, so just to comment on. Any other questions or comments?
Okay.
Thank you.